strange to me. But anyway, I'm Gerry Blobe. I'm one of the lab rats, they like to call me, but I spend most of my time in the lab. And I do half a day a week of GI oncology. And they gave me the nice, I guess my initial topic was signal transduction inhibitors. And I was like, well, that's a pretty broad topic. Then I looked at the syllabus and I was like, well, at least, they've carved out angiogenesis, so I don't have to cover angiogenesis inhibitors. Someone's got that covered. They got someone else doing hormonal therapy. I don't have to cover hormonal therapy. And then they have someone doing monoclonal antibodies for heme malignancy specifically. So I'm not covering those either. So I did want to, as opposed to, we could spend a lecture just talking about all the different molecules, but I really wanted to focus more on the principles of targeting signal transduction, some of the paradigms that have worked, some of the challenges that we face ahead, and try to bring up some examples of where we're looking at the molecular aspects of a tumor and using that to make a rational decision about what to target that with, as well as some of our challenges there. So, trying to get to advance here. So the computer's not allowing me to advance my own slides. Oh, no, this way is working. All right. This is the revised Hanahan-Weinberg review. Back in the original review, back in 2000, they actually attempted to fit every signal transduction pathway into the cell, and it was a mess. And so this time they actually tried to simplify it into circuits and at least make it a little more conceptually easier to understand. Bottom line is, all marks of cancer, you can really boil down those hallmarks down to signal transduction pathways that are regulating those hallmarks. And so what we're trying to do in terms of targeting cancers is then trying to impinge upon those pathways. For the most part, we've gotten really good at working at things that are on and trying to turn them off. One of the challenges that I'll get into is, at least half of cancer is tumor suppressors, and we haven't done a great job in terms of being able to turn tumor suppressors back on, except for some epigenetic regulators that we'll talk about. So that's one of the challenges for the future. And they had this nice little schematic of all the different hallmarks that we have different, for the most part, inhibitors. So we're good at inhibiting things so far. Like I said, we're not good about turning things off. So what are targeted therapeutics? And I talk to my patients about the different treatment options we have for them. We talk about surgery, radiation, chemotherapy, and targeted therapy. They always get really kind of confused by the whole targeted therapy, because it's like, well, aren't you targeting the cancer with that chemotherapy too? They really concerned that, and it's like, well, it's some of it's semantics. I don't really want to get caught up in semantics, because you'll see that some of the things that we think are targeted are not really hitting the target we think they are, and sometimes being precise in terms of hitting a target doesn't make you the most efficacious molecule out there. For the most part, they're at least in theory designed to go after a molecule or a class of molecules as opposed to the entire machinery of the cell. And the categories that they come in: small molecules, antibodies, peptides, proteins, antisense. Those are the things that are the categories that we've been able to achieve. But most of them are small molecules and antibodies at this point. We haven't made a lot of great antisense or shRNA type molecules or ribozymes that are being used in practice. So in just thinking about these in categories, we really are good at impinging upon the interactions between ligands and the receptors, and for that you can either have an antibody that's against the ligand, or you can have an antibody that's against the receptor, or you can actually make a soluble form of the receptor that can then trap the ligand and work very much like an antibody does to prevent that ligand from interacting with the receptor. And then with the cell, for the most part, we're down to small molecule inhibitors. And for the most part, we've gotten pretty good at small molecule inhibitors for kinases, mostly receptor tyrosine kinases, in a couple cases, serine threonine kinases. So we've gotten pretty good at hitting kinases. In terms of the two different types of classes of targeted molecules that have worked pretty well, we've got the small molecule inhibitors that are quite small. Obviously, those can get into the cell. Because they're small molecules, a lot of them were really identified in screens to hit a specific target, but as we know, a lot of these are not very specific. They can hit a lot of different targets, and that can actually be a good thing because they're small molecules that have no effects against the immune system. And the other class is really the large molecules. Most of these are monoclonal antibodies. The thing about them, they're very specific for the target they were designed against, but they can't get into the actual cell, so they have to work at the cell surface. But all have the advantage, and this is increasingly being recognized as an important mechanism of these antibodies of being able to elicit an immune response. All right. So for monoclonal antibodies, we started initially with making them in mice and basically able to make chimeric antibodies. There's still a couple chimeric antibodies out there that we really have to worry about anaphylactic reactions again, like cetuximab. But most of our antibodies are now at least humanized, which really minimizes, and all the antibodies from Amgen are completely human. And then we have a standard nomenclature so you can tell which antibody it is based on how it's named. So in terms of what have we not gotten good at targeting, so a lot of enzymes that aren't kinases. So for example, the GTPases, we really have no good way to target those. We haven't gotten good at targeting transcription factors, things that really rely on protein-protein interactions in terms of mediating their functions. Tumor suppressors are a whole class of molecules that we really haven't figured out how to tackle those at all. And then a lot of other signal transduction that happen based on protein-protein interactions. And I put the BCL proteins up there and G-protein fold receptors. So even though we've made a lot of inroads in terms of how those things work, in terms of actually turning that into a therapy that's being used in the clinic today, we're still stuck. To talk on targeted therapies without bringing up the case study of CML. And this started out with going after BCR-ABL with the chromosomal truncation. And this really was efficient both in animal models as well as in humans to drive this disease. And then going after this with basically a drug screen, high throughput drug screen for things that could actually target the increased activity of BCR-ABL. Came up with imatinib, and this is showing that the relative selectivity of imatinib to BCR-ABL relative to other kinases, so relative to other tyrosine receptors like EGFR receptor and insulin growth factor receptor, they're really inhibited to any significant extent, but PDGFR is hit, for example, c-KIT is hit, and actually one of the fellows in my year training took advantage of that fact to apply this to GI stromal tumors. So this was initially brought to in terms of a high throughput screen. It can do apoptosis. It's really effective. We'll show some of that data. It's also fairly little toxicity compared to chemotherapy. But always surprises me is even though it's fairly well tolerated, it's still about a third of patients who will not be on this therapy and will not adhere to the therapy the way it's meant to, and that's really responsible for a lot of the resistance for patients, and I'll show you some of that data. The other is because it's something that's against a specific target, and because we're talking about cancer, there's a lot of ability to develop resistance, and so there's been a number of mechanisms of resistance that have come up, including mutating the target, so secondary mutations in BCR-ABL, amplifying the target, basically having alternative pathways that duplicate the effect of BCR-ABL even if it is inhibited, and either block the ability of these drugs to get into the cell or increase their efflux from the cell, mostly through P-glycoprotein-mediated MDR. So one of the things that they've also done in CML, and to my knowledge, this hasn't been broadly applied to other diseases, is really have very precise definitions of response beyond in tumors, we're still stuck with 2x2 measurements on a CT scan. Here they really looked at a molecular level, the ability to detect these BCR-ABL transcripts, and looking at actually, we used to just look at the hematological response rate in terms of normal counts. Then we were able to look at the cytogenetic level in terms of Philadelphia chromosome phases. And now with PCR, we can look at molecular responses with a complete molecular response or a major molecular response being the markers. this targeted therapy, for most diseases, looks quite acceptable. 63% of the patients at 8 years are doing well, but 37% of those patients still are not doing great because they either never got to a complete cytogenetic response, or they lost their complete cytogenetic response, or lost it and then regained it. We looked at long-term adherence. So, this is an oral therapy that's fairly well tolerated, but, patients get complacent. They start thinking about this is another chronic disease, and it's the same with hypertension, diabetes. People don't want to treat their disease that they're not having side effects from with agents, even if it's only modest side effects. So adherence to therapy is absolutely critical. Those patients who are able to obtain major molecular responses or complete molecular responses are just directly proportional to their ability to adhere to therapy. So in terms of dealing with resistance, there's been a host of other ABL inhibitors that have come out, and what I think is interesting is both that they've used elegant knowledge of the structure of imatinib with ABL to design therapies that then be able to target those resistant ABL molecules. So nilotinib is an example of that, which basically came out, and it was rational drug design specifically based on the structure of ABL with imatinib. But several of the others, it wasn't that rational. Dasatinib actually was kind of an accidental discovery when they were looking for basically immunosuppressive drugs, and they just happened to find out that it was able to hit BCR-ABL and SRC. And then nilotinib was actually a screen for a SRC inhibitor that actually also turned out to be quite a potent BCR-ABL inhibitor. So both nilotinib and dasatinib are potential first-line therapies along with imatinib, and we'll go into a little bit of the data. And one of the challenges is trying to figure out how to use all these agents. And bosutinib and ponatinib are both used for resistant disease. Some of them do have different bases. For example, nilotinib is not a substrate for P-glycoprotein, so if you have a multi-drug resistant case, you can use that and pass that mechanism of resistance. So there are some reasons to go with different ones, but at this point we don't have a clear answer what's the right way to use all these agents. Now with 5 agents in it, there's actually a couple more in clinical development. So just, in terms of a demonstration of how effective these are, looking at major molecular responses, in the 50 to 70% rate, and comparing that to what you get with imatinib, still pretty good, but clearly nilotinib is a lot better in terms of giving you major molecular responses. And along with that, basically the biomarker for potential response, you see increases and this is a data for dasatinib relative to imatinib. So dasatinib is also a little better than imatinib in terms of these different benchmarks. And then as you'd expect, based on this, look at overall survival and progression-free survival, it does get a little better going on nilotinib versus imatinib. But really nice, overall survival data at 3 years of 94+%. So as a guy who sees a lot of pancreatic cancer, I'm always really jealous when I see this kind of data. But we still have resistance cropping up, and these are the different types of mutations that have been shown to mediate resistance, not just to imatinib, but also to nilotinib and dasatinib. And currently, there's one specific mutation that occurs, the T315 tyrosine, that is resistant to all approved tyrosine kinase inhibitors. We haven't figured out a way to get around this one. So one of the challenges we have with all these agents is really trying to figure out how to use these, and then for these differences in survival, in the next couple of years is going to be coming off patent, whereas nilotinib's got probably another decade of patent life. So imatinib, at today's cost is $8,000 a year, something like that. So it's going to be worth getting that extra couple % in overall survival for that difference in price. It's going to be some of the challenges that we're facing. This is I thought this was a really neat story I saw play out in real life. One of my fellow fellows kind of was looking at He was working on stromal tumors up in Boston because sarcomas are a major health problem in the U.S. At least at Dana-Farber they were. But anyway, he happened to notice that these things had c-KIT mutations, and then he was like for something that would hit c-KIT, and noticed that the imatinib would do it. So he started testing that, and it was pretty amazing because this was a tumor that's very resistant to chemotherapy, and if you don't treat it surgically, most of these patients will die. And was able to see a response rate that was over 50% in these patients, which was just amazing with the pill. So this was really dating myself, but this goes back about 15 years, just an amazing proof of principle that you could use some of these off-target effects to really effectively treat other tumors. And I don't think we're done. A lot of the studies are coming up with a lot of mutations that occur in subsets of tumors, and so we really need to look at that data and think about subsets of tumors that would respond to other agents already out there. I think there are opportunities out there. So this is what we think about in the lab. We don't really want to find the answer. And then the pharmaceutical companies are really trying to turn their diseases into profitable disease. Switching gears and talking about pathways that we've successfully targeted with multiple different agents. So the epidermal growth factor receptor pathway is really pretty complex. There's a number of receptors, the EGFRs, and then the HER family members, HER2, HER3, and HER4. They can work both as homodimers and heterodimers, but they only work as homo or heterodimers. You can actually get different types of signaling out of these, and so you can think about different ways of targeting the EGFR versus targeting the heterodimers with HER2. Most homodimers are going through the MAP kinase pathway and regulating growth, and as heterodimers, you also get activation of the PI3 AKT pathway and regulation of cell survival. And targeting this, like the thematic, we have antibodies that work outside to target ligand receptor interactions, and we have small molecule inhibitors that work within the cell. I'm going to talk about HER2 target agents. Erbitux is one of those early antibodies that's chimeric. You probably have never used it. Has anyone used? Use it in this neck of the woods too often because the. Yeah, we have a special protocol for giving cetuximab. You have to be inpatient and have all sorts of way too many scary reactions in the infusion unit. And there's quite a bit of regional variation in anaphylaxis to cetuximab. In more rural areas it's huge. In New York City it's unheard of. So depending on your rodent choice that you're exposed to, apparently. So the key messages in targeting the EGFR is we're starting to learn a lot, or we've learned a lot in terms of different mechanisms by which you are inherently resistant or could become resistant to EGFR-targeted therapies. So if you have low expression of the ligands, you're less likely to respond to these agents. The big one is RAS mutation status. If you have a RAS mutation, you're definitely going to respond. There are less frequently issues with other forms of RAS. The biggest one most recently is NRAS, which is about 15% of colorectal cancer patients. Apparently those patients don't respond either. Loss of PTEN decreases your response, and activation of the PI3 kinase AT pathway can reduce your response to EGFR targeted therapies. So if you look all together again at all of these, it's probably true that about half of colorectal cancers are not going to respond to EGFR targeted therapies. And since they have significant toxicities, it's important to look for these. Right now, we routinely looking at KRAS status. We're really urgently trying to get NRAS added to that pool because that's another 15% of patients is worth thinking about. And just look at panitumumab or cetuximab. The response rate if you have a mutant RAS is 2%, where if you're wild type, it's up to 30%, and if you look across the whole population, it winds up averaging out to around 20% of the patients, which is what we saw in the initial studies before we knew this was happening. Starting to get into some of the complexities, you think of KRAS as either wild type or mutated, but there's obviously different mutations in KRAS. About 80% of them are codon 12. We know those definitely. If you have those, you do not respond to EGFR targeted therapies. But another substantial proportion, about 18%, happen in codon 13. And there's some reports out there that patients with KRAS, quote unquote KRAS mutated tumors can still respond to EGFR therapies. And it's the thought that it's really these codon 13 mutated patients. But at this point, we do not have enough data to really make a decision. So it's kind of a gray area, and there's different ways of testing for mutation status. So unless you're specifically testing for codon 12 mutations, you really have to make sure that your laboratory can do this well, and you can really understand what the results mean. So not all mutations are the same. So the data that ligands are predictors of response. This is just looking at whether you have high or low levels, and clearly patients with low levels don't respond as well, whereas patients who have high levels have responses, and that definitely translates to differences in progression-free survival and overall survival for these patients. Another marker that has really is not being used yet, but I think should be used, is basically just using the biomarker of rash. That's really part of the side effect profile of these agents. The severity of the rash is really a good biomarker for response to the therapy at the level. So if you have, and this is just looking at across diseases really with rituximab, and showing the severity of the rash does correlate with your response rate and with survival. So since it's so easy to look for, literally all you have to do is look at the patient. You can get a strong indication of, are we really giving these patients the right dose? Should we actually be dosing to rash? That's been looked at, for example, in erlotinib, and there's actually been some studies with rituximab, but it's not currently the standard of care to do that. But it makes a lot of sense to do that for patients. Now, it's also, so with the different target agents in colon cancer and head and neck cancer, we use these antibodies. Lung cancer, where they don't necessarily have amplification of the EGFR receptor, they tend to have more mutations in the EGFR receptor. It's the patients who have these mutations that respond to small molecule inhibitors of the EGFR pathway, like gefitinib and erlotinib. The agents were actually initially approved, but when so gefitinib was initially approved in the U.S., but when they found out there was only a small proportion of patients who were actually going to respond to them, they withdrew the approval, instead of just saying, well, you could actually initially just use them in patients who are known to have the mutation and are known to respond. So it was very interesting seeing that whole story play out. Clearly, there's been new studies that have shown that in patients who have the mutation, you have a very nice response to the small molecule inhibitor, much better than chemotherapy. And so it's now the standard of care for those patients. Again, just like imatinib, there are multiple mechanisms of resistance that have been reported over the last couple of years, with most of those being a second site mutation, similar to what happens with ABL, with imatinib, that alters the ability of these kinase inhibitors to bind. But the other mechanisms are really mechanisms that bypass reliance on the EGFR pathway, like upregulation of the axial kinase pathway that can turn on epithelial mesenchymal transition and activate many of the same pathways. In about 100% of cases, you get amplification of another EGFR receptor, HER2, and then activation mutations or amplification of downstream effectors of the EGFR receptors happens in the rest of the cases. So if you add it all up, we pretty much have solved the problem of how you become resistant to EGFR. And one of the nice things about at least a lot of these studies is that they've done preclinical rationale in terms of, as you'd expect, inhibiting axial kinase or inhibiting PI3 kinase would help restore sensitivity to these drugs, and they've been able to show that happens in animal models. So now it's basically incumbent upon us to basically put these into the clinic in those combinations. So obviously this is not lung cancer in 2103, this is lung cancer in 2013. Hopefully by 2103 that unknown sector will have been filled in by some of you guys. We're starting to slice and dice lung cancer in terms of knowing that. Obviously we have a good handle in terms of what to do with the EGFR mutant population. Also have a targeted agent for the small proportion of patients that have the ALK rearrangement. This was worked out initially at a basic science level back in 2007, which for me is not that long ago. But the data showing that these ALK-positive patients had a pretty good overall survival with treatment with this ALK inhibitor. And then just really, to me, it was amazing, basically within 4 years from showing kind of the basic science arrangement to doing preclinical studies, showing some activity to FDA approval. I mean, it was just amazing. You know, for the VEGF inhibitors, it took about 20 years, you know, from the preclinical observations to actually FDA approval. So I'm encouraged that our timeline to actually get these things in the clinic has accelerated pretty dramatically. All right, in terms of going after another EGFR targeted therapy, the HER2, so there's been of these agents that have been developed, and actually it's pretty cool because a lot of these agents have been developed at Duke or have been shown to be efficacious in studies that have been led at Duke. And I think it was just maybe a year or 2 years ago, Kim Blackwell was giving a talk at one of these big DCI events, and someone asked her the mechanism of action of Herceptin, and she was like, no one really knows. You know, we guess, but there's a bunch of potential mechanisms by which it works in terms of causing internalization of the receptor, blocking the receptor from interacting with its ligand, potentially the immune-mediated interaction that we now know a lot of these antibodies have. But we do know you have to overexpress the target. So we at least go after HER2 overexpressing cancers, breast cancer, and now also gastric cancer. In terms of other, besides Herceptin, Lepanid was developed, goes after HER2 and EGFR, a small molecule. There's a pertuzumab that goes after the ability or the requirement for these things to homodimerize to be effective. So it actually interacts with the dimerization domain on HER2 to block dimerization. And then most recently, TDM1, which basically uses a Trojan horse approach to target two overexpressing cells with basically an antibiotic, an anti-tumor agent. So, you know, again, all these agents is a question. So, you know, if you look at the NCCN guidelines, we have choices in terms of either using the dimerization inhibitor or straight Herceptin in the first line with chemotherapy. Those patients have already been exposed to one of these agents, usually Herceptin. Then we have choices to use the TDM1, the Trojan horse approach, or use a small molecule inhibitor, or use a Herceptin again, but with other chemotherapy agents, or to use a combination of these agents together. Really, you know, it's a pretty complicated landscape, and there's not just one right answer for these patients. Just some of the data that led to some of those recommendations. This is the Cleopatra study that looked at trastuzumab versus the combination of trastuzumab and pertuzumab, and showed that the combination was clearly better than single agent alone, so thus putting a combination therapy as one of the options for these patients. The Amelia study that looked at basically TDM1 versus lapatinib in patients, and again showed increased in overall survival with TDM1 versus this combination therapy. And other tyrosine kinase inhibitors, so we have a number, and some of these are touched on because they go after VEGF, so you may have heard about them when, who talked to you about angiogenesis? Was it Herb? Or has it not happened yet? Hasn't happened yet. Coming future attractions. Okay. So a lot of these tyrosine kinase inhibitors go after a lot of targets simultaneously. So the top two basically can go after VEGF, but also PDGF, and both have been approved. Renal cell carcinoma as well as sunitinib is also used in GI stromal tumors that have progressed on imatinib. And erlotinib is just another small molecule inhibitor that's used along with gefitinib in second-line lung cancer and a little bit still in pancreatic cancer. In pancreatic cancer, it's kind of interesting that this is another disease in which dosing to rash seems to make a difference. So if a lot of patients with lung cancer and pancreatic cancer tend to smoke, and tobacco exposure definitely decreases your ability to metabolize, so you tend to have much lower concentrations of this drug. So dosing to rash might work in those patients. And then there are a couple multi kinase inhibitors that are used for metastatic medullary thyroid cancer. It can go after VEGF, but also EGFR, so it's really not known how, what target's really important, although RET is thought to be one of the key ones. And then vemurafenib, which goes after BRAF, specifically for BRAF mutant metastatic melanomas. So, tyrosine kinase inhibitors, we're able to target mTOR, which is a serine threonine kinase. Most of these really initially came after the observation that we could use mTOR inhibitors as an immunosuppressant agent, but analogs of these were tested. Renal carcinoma was an obvious choice to start these because there was a lot of preclinical data showing that in a HIF-dependent manner you had anti-tumor effects with these agents, and in two of these, everolimus or Afinitor and temsirolimus have been FDA approved for renal cell carcinoma. These are both active site inhibitors of these serine threonine kinases. Is this really targeted? So proteasome inhibitors, pretty much every protein that gets internalized in the cell is degraded by the proteasome, and so there's huge number of targets for these. And I would have been the first one to be surprised that proteasome inhibitors could be at all tolerated by individuals, let alone effective for cancer. But I'm the one that bought stock in these. So Velcade is the first one of these for multiple myeloma, and there's another one that's also been approved for refractory myeloma. Both of these are thought to work at least in part through going after the NF-kappa B pathway, and both are active site inhibitors of proteasomes, usually the 26S proteasome. Our best shot on goal in terms of going after tumor suppressor genes is epigenetic regulation, so HDAC inhibitors, and really going after the ability to activate genes and thereby having effects on differentiation, potentially having effects on apoptosis and cell cycle progression. And at this point, there's just two HDAC inhibitors, vorinostat and redempsin, that have been approved for a particular subset of T-cell leukemias, lymphomas, sorry. And then really not necessarily in the category of, very targeted is thalidomide and lenalidomide, which have multiple mechanisms of action, some of them thought to be anti-angiogenic, but also modulating the immune system, as well as potentially having direct anti-tumor effects, but have been approved and have really made progress in both multiple myeloma and MDS. And another antibody that's against ipilimumab, which is basically helps increase cytotoxic T cells, TLA4, their function, and is indicated in metastatic melanoma, and has been the emergence of target therapies in melanoma the last couple of years. So I want to spend the last couple of minutes just talking about if you have an embarrassment of riches in terms of different agents that all kind of match up pretty well in terms of progression-free survival and response rate and overall survival, and then basically also in terms of their toxicity. A lot of times we, in terms of chemotherapy, if we have equally efficacious agents, we kind of try to look at the toxicity profile and, does the patient want to have neuropathy from oxaliplatin, or do they want to have diarrhea and hair loss from irinotecan? We try to make choices based on side effect profile. But if side effect profile looks pretty similar, then how do we really choose, which agents to expose these patients to? And the bottom line is we have to start looking at the there's a second line also looking at choices in terms of whether to go for tyrosine kinase inhibitors or mTOR inhibitors in these patients. Again, the efficacy data is fairly similar. When you look at the management, one of the things we do is some of these actual drugs were approved in different settings. So for patients who are at poor risk up front, we tend to go with the mTOR inhibitor. But for the patients who are more favorable risk, start with the tyrosine kinase inhibitors and then tend to use mTOR inhibitors in the second line. But a number of studies that are going on where they're basically looking at what sequence to use these. And this is challenging because all of these studies, when you look at all these targeted agents, how many of these different studies can you do? Looking at agent A versus agent B versus B, it just gets pretty complicated, but that's how we're trying to figure out how to sequence these agents correctly. So, and we're trying to match these with personalized medicine, getting increasingly sophisticated in terms of how we can look at tumors from just looking at protein expression to looking at gene copy number to looking at all the genes and matching up those, and then with the whole genome atlas, really be able to get a really first look at the sequence of these different cancers. But we see a lot of challenges in terms of defining new therapeutic targets. I mean, I think we know most of the pathways that are important, but really knowing which ones are going to be important for specific cancers. Confirming that we're actually inhibiting the target within cancers. For a lot of these cancers, we have challenges in terms of being able to get biopsies before and after treatment. It's very straightforward in hematological malignancies, but much more difficult for a lot of solid tumors. Getting better in terms of determining who's going to respond to therapy and figuring out better preclinical models to rationally combine and sequence all of these target therapies is really, and I think one of the best things that we could do is really figure out a better preclinical model for figuring this out. And one of the ways forward that I think is pretty promising, and she's doing quite a bit of it, is doing these patient-derived xenografts, where the patient-derived xenografts, which have never been passaged on plastic, passaged in living mice, tend to more realistically mirror what's happening to the patients. And that way we can really at least start modeling it. It might be, but that happens, can work for those patients who have enough of a tumor to be able to take out and stick in a mouse. If you have a very limited specimen to work with, that's going to be difficult. And then a big question that maybe Zafar will come talk to you about is how can we afford to treat patients with all these therapies? The ABL inhibitors, there's a lot of data. If you use 2 of the inhibitors, you decrease the emergent resistance and you increase the response rate even further relative to any one agent alone, but now you're talking about 2 expensive therapies at the same time and for a long period of time. So these things can get quite expensive. So we do have to figure out how to afford treatment. So I read it very interesting. I didn't really realize this, but imatinib was actually at about a third of its current price, and then they tripled the price based on the competing, was it interferon, was basically that they were benchmarking it against. They basically just decided this is what the market can bear. I don't think that's going to happen too much longer. So 4 years ago I gave this talk, and I was talking about target therapies are emerging, and so I think at this point they have claimed their spot as one of the backbones of therapy. I already brought up the really preclinicals are key, and I like what has been done in CML in terms of looking for activity of agents, but we really need new paradigms in terms of looking at these in more tumor types, and then increasingly patient selection, matching up specific mutations with specific agents that are going to target those. It's going to be kind of standard of care going forward. What I got for you guys? Questions?
I was just looking at the question about resistance and how they determine whether someone's resistant or not.
Right.
And just as a example, I'm looking at KRAS mutations. This is a test by Quest Diagnostics. They've identified that, there are some that have a high likelihood of being significant if they're in, exon codons 12 and 13. So
yep.
They do a PCR, they sequence exon 1 and look for those. The question I have is, how to determine, I mean, are all those mutations the same?
No, they're not, and that's what that one slide was getting at. So for the most exon 12, a mutation exon 12, no one's shown that, shown that does not. So data points to those being significant for lack of response. It's exon 13 that's the question. So it's about 15% of patients, and patients, first of all, it's not as oncogenic as exon 12 mutations, so it really isn't thought to be as big a driver of the tumor to begin with, and it's really not thought to be as predictive of lack of response to therapy. That's the way of thinking about it. These are really not predicting who's going to respond or predicting who's not going to respond. But those mutations in 13, codon 13, do not predict as strongly as those in 12.
So does it still come up as positive if they have a mutation that does?
Well, that's why you really have to dive in. I mean, we should do our own in-house testing for KRAS mutation status so that we can get that granularity. But if you just send it off to Quest and you just get oh yeah, it's mutant, but they don't tell you what their definition of mutant means, then you could be not exposing someone who would benefit from that therapy,
right.
And leaving them a shot.
Give a EGF agent to somebody with a codon 61 mutation. Like if they one that was a little bit more questionable, would you still consider giving?
Still consider it, and I think that's where it becomes a conversation with the patient, letting them know, like, hey, listen, we don't definitively know the answer, but you at least have a shot at benefiting from this, and these toxicities, and are you willing to go for it? So this whole NRAS story just came out in New England Journal the last couple of weeks, and so we're really scrambling with my dad on molecular diagnostics to get an NRAS panel up and going, but it takes time because he's not only going to be able to do it, he's got to be able to do it reliably so that we can actually trust the answer and make a clinical decision based on that answer. So just being able to operationalize a lot of these takes a lot of time, and so the implementation in patients lags well behind actual science.
At this point, multiplex them, or do you need more tissue to do, KRAS than do NRAS, or can you just do the same just multiplex?
I think you'd be able to multiplex, but again, you do work. I mean, these are not the types of things where you want to be doing all the amplification, right, and introducing, error rates and all that. So you want to. So that's part of the challenge too. For most, cancers, there's a lack of tissue in which to do this with. Again, it gets down to cost. They want to make sure, the university wants to make sure that they're going to get paid for doing these tests. There's lots of things we could be testing for, some things that are obviously actionable. There's other things that would be nice to know and at some point could be actionable but aren't currently. We'd like to be looking at all those right now and trying to figure out, well, is there some other market resistance or sensitivity to things that we're already using, and use the EPIC to capture that data. It's the only good what I can say about it.